Hi guys, it is week 6. It's Tuesday, Endocrinology. We are going to get started finally with Cushing's disease, but we've got to talk a little bit about gluconeogenesis. So kind of a short lecture, I'd like to think. So I do hope you've remembered glycolysis, gluconeogenesis, and the Krebs cycle. Now I cut a lot of stuff out of here because I was getting out of control because this is not biochemistry, but I do want you to know this kind of watered down version of this. So you got to understand gluconeogenesis to understand what one of cortisol's mission statements is. And one of its mission statements is to make sure the blood has proper levels of glucose under stressful conditions and during fasting conditions. And it can do so by breaking down things like fat and breaking down muscle protein and even using lactic acid to make glucose via gluconeogenesis. So let's see how that all works out. So muscle proteins, of course, as I just said, cortisol will cause muscle protein catabolism, and it'll break down muscle proteins into free amino acids which get in the blood. It has some other functions as well. I have a slide at the end that shows kind of a summary. But cortisol also discourages muscle cells from uptaking glucose, because remember, and uptaking insulin or binding insulin, because we want to leave glucose in the blood, so we don't want muscle cells sucking up blood glucose. So there's other mechanisms, but the one I'm worried about here is catabolism. So we're actually breaking down muscle fiber, and that's going to dump a whole bunch of free amino acids into the blood. And most of them end up in the liver cells, where they can be converted into intermediates of the Krebs cycle. And this is why that in biochemistry you had to learn glycolysis and you had to learn the Krebs cycle. And now you have to, well, if you know glycolysis and the Krebs cycle, you almost know gluconeogenesis, because it's just running stuff backwards through those cycles, as we'll see. The goal of amino acid, the goal of every amino acid that's floating in the blood is to become a glucose molecule or help make a glucose molecule. And the short-term goal for most of them is to become oxaloacetate, which is the end product of the Krebs cycle, if you'll remember. So here's a cartoon, and I used to have all the names of the amino acids, but that's beyond me. I don't think for this class you have to know that, but you do have to know that most of the amino acids, with the exception of leucine and lysine, they cannot be used to make glucose. And ultimately, I cut some of this out so we can see it. But up here is glucose. Let's see if I can. I write so slow. I need like one of those pencils. Anyway, that's kind of where we're going. But you can see that most amino acids, these are the intermediates of the TCA cycle. Recall that acetyl-CoA, or recall that from glycolysis you make pyruvate. Pyruvate is converted into acetyl-CoA, which is converted into citrate by bonding oxaloacetate with acetyl-CoA. And then it goes to isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and then into the star of the show here, oxaloacetate. But notice these amino acids, and we won't worry about the enzymes or how this happens, but they can be converted into alpha-glutarate or alpha-ketoglutarate. And these group of amino acids, and there's probably 5-6 of them, that can be converted into succinyl-CoA. And there's more of them can be converted into fumarate, and then some into oxaloacetate itself. And then there's others that can be converted into acetyl-CoA. And there's a couple of them that can be converted right into pyruvate. So that's the deal. But the key is right here. The goal is to become oxaloacetate. That's the end product of the TCA cycle. We can grab oxaloacetate by using and by applying the enzyme PEP carboxykinase. We can create phosphoenolpyruvate here. I'm getting ahead of my slides. But once you do that, see, these enzymes are all two ways. So the enzymes, for example, that's used to glyceraldehyde 3-phosphate. An enzyme is used to convert that into phosphoenolpyruvate. See, these are all two-way enzymes, so the same enzyme can be run the same way, so you don't have to memorize any more enzymes. But phosphoenolpyruvate, as it converts into pyruvate, it can't go backwards. That's one of the irreversible steps. So pyruvate has to be kind of routed around in a different way, but we'll get to that in a second. But for right now, this slide, note the amino acids can go into many of the intermediates of the TCA cycle to ultimately make oxaloacetate, which can be run through gluconeogenesis. Got it? Adipose tissue or fat, that cortisol binds to fat and it causes the breakdown of fat. Remember, fat is AKA is triglycerides or triacylglycerides. And glycerides, that's messed up there. Try to remember to fix that. Triacylglycerides. And it can be, the process of breaking down fat is called lipolysis. So cortisol stimulates lipolysis. What is fat made of? Well, fat is made up of a glycerol molecule and 3 fatty acids. So that's what it breaks down to. The glycerol is ultimately converted into an intermediate that you may remember of glycolysis called dihydroxyacetone phosphate. And that can be run backwards to make glucose, backwards through glycolysis pathway. Or, I mean, when you run backwards through glycolysis, that's gluconeogenesis. Okay, there's a couple different enzymes you have to use, but you shouldn't say running backwards through glycolysis, even though that's what we're doing. You should say running through glycolysis. And we'll look at a picture of that in a minute. These free fatty acids can't be turned into, there's no way to turn these into glucose. However, these free fatty acids are absolutely crucial for the process of gluconeogenesis because they're burned. Two units are chopped off the fatty acid tail, and each turn of some enzymes in a process called beta oxidation. And beta oxidation basically creates NADH and FADH, which is converted into ATP. And so we take this ATP, and that's what fuels the reverse steps of the glycolysis, which I'm not supposed to say, but fuels the steps of glycolysis. So without fatty acids, you can't run glycolysis, so they're not wasted. But here's the board question, or Dr. Dumanier's question: they cannot be used to make glucose in and of themselves directly. Indirectly, yeah, because this is where the energy to power that reaction comes from. Got it? So here it is over here. So fats, and there's cortisol splits fat into fat molecules into glycerol. Fatty acids are used to make tons of NADH, FADH. I didn't put that in here, but ultimately it's made, and this arrow is actually wrong, right? It should be used, well, it's just used everywhere in this whole process, but it could also be used up in here. It's used to run this whole process backwards. So that's where it comes from. But glycerol is right here. So the first step, glycerol kinase, and it's really bad to have mutations in glycerol kinase because you need that to make glycerol 3-phosphate. Glycerol 3-phosphate via glycerol 3-phosphate dehydrogenase is into a very familiar enzyme. You had or intermediate of glycolysis. You had to memorize a DHEP, dihydroxyacetone phosphate, I'm sure. And then there you go, off to the races. We just run backwards, 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 and there's glucose. Although there's 2 enzymes, new enzymes that we need. Try not to get ahead of my slides. What about lactate or lactic acid? Even that's not wasted. Now this might be too complex for chiropractic boards. I'm not sure if Dr. Dumanier taught you this or not. But there is a Cori cycle that lactic acid, aka lactate, can be run through to create pyruvate. And once pyruvate is made, you can ultimately get that to oxaloacetate, and you're off to the races. Okay, so what do we do with oxaloacetate? So now's the time where I tell you about this. So oxaloacetate is converted into phosphoenolpyruvate, by a very important enzyme called PEP carboxykinase, or PEP stands for phosphoenolpyruvate carboxykinase, and that's a mouthful. And you lose a CO2 molecule in this process, but you actually convert oxaloacetate into PEP. And then you just run glycolysis backwards, which I'm not supposed to say, but you do. There's two more enzymes you need, and you can run this thing right backwards, no problem. Okay, so here's kind of the pathway here set up, and here's these two new enzymes. So we're talking about making PEP, and that can be converted into glyceraldehyde 3-phosphate. And I didn't write the enzymes in there, there's no room, but you already memorized those for Dr. Dumanier. That can go into fructose 1,6-bisphosphate, and that we need a new enzyme. I can't remember the enzyme off the top of my head, but I think it was fructose 6-phosphate dehydrogenase to get to fructose 1,6-phosphate. But the bottom line is the glycolysis enzyme cannot be used. We need a brand new enzyme, and that is called fruct 1,6-bisphosphatase, right? Or fructose, that's short for fructose 1,6-bisphosphatase. So that's one of the irreversible steps in glycolysis. And then the very last step, remember the trapping step, where glucose is trapped inside a cell from this first step in glycolysis. Glucose going to glucose 6-phosphate. That's hexokinase. There's two enzymes depending on what tissue, and one is hexokinase. But it doesn't work. It's an irreversible enzyme. So we need glucose 6-phosphatase. So you should be able to know these and pick these enzymes out and know that they're not part of the glycolysis pathway. They're part of gluconeogenesis. They're not two-way enzymes. You should know that about them as well. Right? Another enzymes I didn't mention here, but these two enzymes are also part of the gluconeogenic pathway. We have one called pyruvate carboxylase, and you can take pyruvate, is interesting, and you can run it directly into oxaloacetate by adding a CO2 and with this amazing enzyme pyruvate carboxylase. That way you skip going through the TCA cycle. If you want to go through that, that's fine. But this one can take pyruvate and convert it flat out into oxaloacetate. So that's interesting. And then the other enzyme we need for gluconeogenesis is the PEP carboxykinase, which we talked about. So if I asked you, you should be able to pick out the 4 enzymes of gluconeogenesis, the four enzymes that are different compared to glycolysis. And that's pyruvate carboxykinase, PEP carboxykinase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. Got it? And fun facts, if you run glycolysis anaerobically, remember you yield lactic acid or lactate, and again we can convert that. I think that's so cool. I didn't know this when I went to school. This Cori cycle wasn't around, or at least I didn't know about it back then. So I was amazed when I started studying this stuff to put this in. The Cori cycle takes lactate and it makes pyruvate, and then you can use PC and can make oxaloacetate. Got it? Everybody good? And here's that kind of overview slide, which is really easy. I can make several questions out of this. But muscles, so cortisol hits muscles and it causes protein catabolism, or you could say degradation, releases amino acids, but it also decreases protein synthesis. So if you're a bodybuilder, this is not good because you can't build muscle. It inhibits that. And the muscle cells, it discourages them from using glucose. You can't bind glucose because you can't bind insulin. You need insulin to bind glucose and take the glucose inside the cell. So it does everything it can to keep glucose in the blood, right? And then it gets into the liver, and we know what happens through glycolysis. You make glucose. It also binds. We've covered this. It binds to adipocytes and causes lipolysis, and that releases glycerin or glycerol, and we said that can be converted directly, or it can be converted into glucose in the liver. But also the free fatty acids via beta oxidation, that's where the ATP comes from to power gluconeogenesis. Got it. Are we good? All right. Let's see what else in here. Cortisol inhibits the synthesis of glucokinase in the liver cells. Okay. And that discourages the trapping of glucose in the liver cells. It stops glycolysis. Yeah, that makes sense. Glucokinase is that first step, right? So that's another thing it does. Hexokinase and glucokinase are both first step enzymes. It takes glucose and makes them into glucose 6-phosphate. So it's in liver, you have glucokinase is the enzyme there for that first step of glycolysis. And cortisol will block this so you can't trap glucose. We're trying to make glucose. You don't want liver cells taking it in. Okay. Juicy slide there. Okay, Cushing syndrome and disease. So we'll get started on that. Cushing syndrome, also called hypercortisolism. The S, I think I've said this before, but the S, here's how I remember it means something. Because Cushing's disease and Cushing syndrome are not AKAs. They are different. Cushing syndrome means something is causing the release of cortisol, right? So S for something. There's many different things that can cause the excess of glucocorticoids in the blood serum. So this is the major category here. This is a potentially fatal disease. In fact, the 5-year prognosis, 50% of people will be dead if this is not properly managed. So this is a serious disease. There's many possible causes, but you can break them down into 2 camps. ACTH-dependent causes, meaning it depends on ACTH is the problem, or ACTH-independent causes, which means ACTH is not the problem. Let's look at ACTH-dependent causes of Cushing syndrome. And this means that the disease is driven or depends on an overproduction of ACTH. That's the problem, an overproduction of ACTH. So yeah, that's it. So something's way overstimulating POMC, the conversion of POMC to ACTH. Maybe it's, we'll look and see what it is. But yeah, that's the deal. Bottom line, there's nothing wrong with the adrenal gland. There's no tumors in it. There's no cancer in it. It would be working fine. But there's just a problem with ACTH. There's too much of ACTH. We talked about too little ACTH. Now we have too much of it. So what are these somethings that can cause ACTH-dependent causes of Cushing syndrome? What's the number one? This is where the confusion is. The number one cause of ACTH-dependent Cushing syndrome is Cushing's disease. Cushing's disease. Cushing's disease means tumor. So if you have Cushing's disease, you have a tumor in your anterior pituitary. And that it's a corticotroph adenoma typically. Now cancer can do this, can secrete ACTH as well, but it's typically a adenoma, which means a tumor of glands, and specifically they're made of corticotroph cells, which do what corticotroph cells. They secrete POMC, which is converted into ACTH. And these tumors don't have receptors for cortisol, so you can't turn it off by that double negative feedback system. All right, so Cushing's disease, everybody repeat it after me. Repeat after me. Cushing's disease is the number one cause of Cushing syndrome, of any type of Cushing syndrome. That's by far the most common. All right, there are some other ACTH-dependent causes. So we have ectopic ACTH secretion. Maybe you have a tumor of corticotroph cells that are in the pancreas. Well, that would do the trick. This stuff is rare, but it could happen. So yeah, that can do the trick as well. Ectopic means outside of the normal place where it's supposed to be secreted. How about the ectopic release of CRH, right? Or how about, yep, so, I mean, just how about a tumor in the parvocellular neurons? I mean, that could do the trick as well. That wouldn't be, Goodman calls it ectopic, but I mean, it's not really ectopic if it's releasing CRH from the hypothalamus. But you can have ectopic CRH releasing tumors from other places as well. So an adenoma made of parvocellular neurons can, if it's in the pancreas, you got ectopic corticotropin release, corticotropin releasing hormone, or CRH. We all know what CRH does. It's a hormone, so it can still get into the anterior pituitary and cause the release of POMC/ACTH. And yeah, could be caused by a CRH tumor anywhere in the body. I just caught that. That shouldn't really call. They should make 2 categories for that. I'll try to go fix that. That's the way Goodman had it, but he needs to fix that too. Let's see. DHEA elevation. Well, yeah, if you have an ACTH-dependent Cushing's disease, since ACTH is driving stimulation of the fasciculata cells, which release cortisol. Remember, ACTH also stimulates the reticular cells, which make ADA, or which make the DHEA, or you could say, I don't know how this is going to show up on boards because they don't like abbreviations, DHEA. I have a feeling they might say the adrenal steroids, or sorry, the adrenal androgens, or they could say DHEA is converted into androstenedione. So you definitely need to know all those 3. Androstenedione, DHEA, or the adrenal androgens, because I'm not sure which one they're going to use. I haven't been on that part of the test writing committee yet, even though I requested. Been on all anatomy and embryology and such. Anyway, so what? Well, if it's a female, it causes something called adrenal virilization. Adrenal virilization, and that's where, the same like a female bodybuilder who takes testosterone to get her muscles big, same thing. You're going to have male characteristics. So you get facial hair, you get a deep voice, you get acne, you'll start losing hair, you can go bald. You're not going to go like bald, but you can start getting your hair thinned out. Increased libido, increased muscle mass. Those are signs of adrenal virilization. Males can have it too, but females it's more pronounced. I'll say now what about ACTH-independent causes? So this excess secretion of glucocorticoids has nothing to do with ACTH. So ACTH-independent causes. So the serum cholesterol has nothing to do with ACTH. So there's nothing wrong with the pituitary. There's nothing wrong with the hypothalamus. The problem is the adrenal gland itself. Got it? Specifically, it's a problem usually with the adrenal cortex, but the zona fasciculata. So ACTH-independent causes, what are some examples? Well, we could have a unilateral adrenal adenoma or a carcinoma that lives in the zona fasciculata. And so if it's a tumor, and if it's an adenoma, that means it's a gland tissue tumor which is capable of secreting. And if it's made of fasciculata cells, it can secrete cortisol, and that's exactly what happens. Cancer can do that as well. There's a condition called PPNAD, called primary pigmented nodular adrenocortical disease, and it's about 2% of all of the ACTH-independent Cushing's disease causes. Another one I gotta fix. Yep, so that's another one. McEwen-Albright syndrome and iatrogenic. So this is common. So if you take too many steroids, if you have really bad RA and they keep raising your corticosteroids up, you are going to have ACTH-independent Cushing's disease. And that would be from exogenous use of glucocorticoids. How about some other causes? Alcoholism. So this is interesting. Alcohol can bind to parvocellular neurons and cause them to release their juices, which is CRH. And we all know what CRH binds to corticotroph cells and stimulates the release of POMC, which is converted into ACTH lickety split. And yeah, so that's drinking a lot of alcohol is a problem. All right, so that's it. That's kind of it's short lecture, but it's juicy. I mean, that biochemistry stuff, I do expect you to know it to the level I taught it. All right, see y'all later.